they actually print out the names of the amino acids, like phenylalanine, leucine, and serine, and all that. And what they're doing, and this is not something you need to worry about, but it's kind of neat the way they did it. And under each of the amino acids, there's kind of a little, it's like a little rectangle. That represents the same thing for each amino acid, just for your interest. The way amino acids are, when usually in a class, when I talk about an amino acid, I'll draw it. Like this. So this is quote a typical amino acid. But what we're really talking about here is, in terms of amino acid, this is the acid group here. I'm not going to test you on this. You don't need to worry about it. It's kind of neat to know this. This is the acid group. This is the amine group. And the word amine means it has a nitrogen in it. So all amino acids have an amine group and an acid group. That's why they are amino acids. But this little thing, if you look on the periodic chart, you won't find a plain old R.
No.
It's a side chain. And this is what makes each amino acid different from each other amino acid. It depends on what is in the place of that R. There is no such thing as R. It's just a symbol standing for something.
It's
The
just
simplest amino acid has a hydrogen right here. The next simplest amino acid has a carbon with hydrogens. And then it can get more and more elaborate. Several carbons, you can have rings of carbons and all that. And even some nitrogens in the side chain. So what they're showing on this chart, the way they've done it, it's kind of a neat way, just a simplified way, but they have that little kind of a rectangle underneath each amino acid. That's meant to be the part of the amino acid that's similar in every amino acid, including this carbon right here. What they're showing above that is the slight differences. You can't really tell. They've done it in a very simplified way, but the slight differences in each of the amino acids. It's not a big deal, but it just kind of gives you some idea of what it looks like. So I think that's kind of a neat way of doing it. The other thing is you can see how some of these amino acids, there's several different codes for it. For example, leucine, there's 6 different codons that code for leucine. Here's serine, there are 2 up here, and there should be 4. I think here's another 2 here. I thought there were more than 4, but maybe I can't remember right. But anyway, there's at least 4 for serine. Some, so they often use the word, it's a degenerate code. Now you have to be very careful when they use a word, and it's an English word that they're using in science, because it may not mean the same thing. So if you talk about a person being degenerate, that means they're probably over on 2nd Street passing out half the time, and they're getting picked up from the tank. That would be a degenerate. Okay, but this is when they say degenerate code. What they mean is it's not a one-to-one code. For any one amino acid, there's at least 1 codon that codes for it, but there may be more than 1 in some cases. In some cases, there may be 4. So it can vary a lot. And that's all they mean when they say degenerate code. It has nothing to do with its moral character. It's actually a very nice code. It pays its taxes, whatever else. Nice. So anyway, okay. So let's that was just a quick thing. But do you remember how to use this? Did you know the deal on this? The genetic code is these codons.
Mm-hmm. Well, it didn't look like this.
Yeah, but this one,
It's the same code, though. It looks, if you get out the other code, you can look at it, it's the same. Nothing's different, except the way they've done it. Just to make it look a little easier to read. First position, second position, third position. And the codon, what is a codon? Let's remind ourselves of this. A codon.
Codon is this.
I tend to put it in capital letters, you don't have to. There's 3 bases on what molecule? Remember? Messenger RNA, right? mRNA. 3 bases on messenger RNA that codes for how many amino acids? 1 amino acid. So when you look at the code, so let's just pick any code. You know, you say GAC or something like that. So you say, let's just say GAC. I didn't even remember which one that's codes were. So you look down here, first position, you look to the first one, you look down here, G's at the very bottom. Then you say, what, the second position is A. Here's A over here, so it's going to be one of these two. And then C is the,
Oh, neat.
So it's GAC, and it says it is aspartic acid. Yeah, so this goes for aspartic acid. And the way they've done the chart, the way I like it, is so it's kind of neat, is that you can look at this and you say, hey, look, there are two different codons that code for aspartic acid. It'd be GAU and GAC. And if you go right below that, GAA and GAZ both code for glutamic acid. So I just think it's a little easier to read than the one that's on the lab, your lab.
It cheats you to the
what lab number was this? Lab 11, I think it was we did last week.
Arginine.
Same code, they haven't changed, it's the exact same
No,
code.
I agree.
Nothing's changed, it's just the way it's presented here. I think it's easier to read. It was actually nicer in the book because it was in color. You know, and so it did a good job on it. But yeah. Okay, any questions on the genetic code?
No, I get it now.
One thing we didn't get to last time, I said I wanted to talk about were mutations. And then we're going to get into mitosis. Okay. Oops. So I didn't write down. Oh, I didn't write down. Behind here. Sorry. Mitosis and the cell cycle. Oh God, I've got a lot of terms to go over. Mitosis and the cell cycle. I'll tell you what, I want to put the terms up as we use them. Let's just wait a second. Let me talk real quickly about mutations, and so I can just quickly review some terms that we need to use anyway today. The clean. So let's remind ourselves, I'll leave that word up because that's a good word. Chromosome. What is a chromosome? Now, to be honest, and if you want to be really technical, a chromosome is what you see in the microscope when DNA is highly supercoiled. Okay? But we'll still use the term anyway, whether it's coiled or uncoiled, you know, because it's so much easier. So what is a chromosome? It's a long strand of
DNA.
DNA is always a double helix, right? So it's a long strand of DNA.
Unless it's a virus.
DNA. This is in eukaryotic cells, and let's remind ourselves, eukaryotic cells, meaning our kinds of cells, plant cells, algae, not bacteria, everything but bacteria. We have pairs of linear chromosomes, and linear just means if you stretch it all out, it'd be a long straight line of it, as opposed to prokaryotic cells, bacteria. They have one circular chromosome. I'm going to put this in parentheses because mostly what we're going to be talking about is eukaryotic cells, and certainly when we do mitosis, we're only talking about eukaryotic cells since bacteria don't do it. Do it this way. No, we'll get that in a second. That's for future. Just hang on to it. So, okay, so long strand of DNA. We have pairs of linear chromosomes, and when I say we have pairs of them, you know, We have 46 chromosomes. Humans have 46 chromosomes. 23 pairs. Let's actually write this down. And of those pairs of chromosomes, each pair, we got one member of the pair from our mom, one member of the pair from our dad. Okay? So let's do this. Humans have 46 chromosomes. But again, what we're really talking about, 23 pairs.
Mhm.
Okay, 23 pairs. One member of each pair from our dad, one member of each pair from our mom.
All chromosomes come in pairs, right? If you get singular, you've got a problem.
Well, yeah, and we'll get to that in about 4 lectures from now. Yes. Yes, too much. It turns out for something like mammals, where everything is so highly regulated, too much genetic material is bad, too little genetic material is bad. It seems to mess up
There's
all kinds of regulation, get all kinds of problems. And we will get to that.
there's
So humans have 46 chromosomes, 23 pairs, one member of each pair from mom, and the other one from dad. Okay. Now, chromosomes, long-stranded DNA, but we usually talk about segments of it that code for for a protein. And what do we call those segments? And I just said the word, didn't I? Genes. Okay, so we said a gene is the length of nucleotide bases, length part of a chromosome. However we want to say it, we'll say a gene is the part of a chromosome that codes for, has the information for one protein. And remember, originally when they said it, they said one gene, one enzyme. And then we said one gene, one protein. You could say one gene, one polypeptide in some cases. And there are other variations on that. But one gene, one protein is the easiest way to say that. One gene, one protein. And remember, I'm not saying these are equal. A gene doesn't equal a protein. No. They're totally different. A gene is nucleic acid material. A protein is amino acid material.
Yeah.
So they're totally different kinds of molecules. But what we're saying is, what is a gene? A gene is the information, the genetic information, the DNA, that codes for one protein. Right? Question?
You're not saying that each individual gene is responsible for a single protein.
Yes, I am. Responsible for giving the information to make one protein. Yes. And remember, we talked about this last time. How many genes do we have? And they used to think 50,000 to 100,000 genes. And when they sequenced the entire genome, it came out to be around 29,000. They go, that's not enough for humans. Doesn't seem like it should be enough. Because, as I told you, fruit flies don't have too many less than that. We think we're so much above fruit flies.
It's just how it's put together.
It's what the genes are expressing, obviously, and it's certainly enough to make a human. And just because a fruit fly might have, I don't remember how many genes a fruit fly has, but it has many thousands, but I don't remember the number. Just because a fruit fly has, not too many fewer than us, that doesn't mean that, so how the genes are expressed, how they interact, what they do. We're human. That's the important part, and they're fruit flies. That's the important part for them.
They know how to function properly.
So the number of chromosomes or the number of genes isn't the important thing. It's what how everything's expressed. So what about a chromosome? Now I can say a chromosome. I'll say a chromosome is a linear array of genes. So how many genes on any one chromosome? Well, that's no way to say. It depends upon the chromosome. And some of our chromosomes, for example, are very big. Some are sort of small. The big ones have thousands of genes on them. If it gets too cold, shut the door. So, you know, I mean, the smallest chromosome that we have, or males, I should say, is the Y chromosome. It only has, I don't forget how many genes, something like 30 or something like that. So it's a very tiny little chromosome. The X chromosome has thousands of genes on it, you know. So anyway, all right.
The X just changes a couple things.
So let's go back. Let's go back and talk about this word of mutation. What is a mutation? All right, a mutation is a change in the genetic, well, in the sequence of DNA bases on DNA that causes a change in the amino acids that we make. Let's write that down. So, you know, you have a gene. A gene then is so another way to say a gene. Let's say a gene. A gene is a linear array. Or sequence. I said array. Sequence. I'd rather have sequence. Let's change that word to sequence. I think that's a better word. And I think I'll change it down here too. Linear sequence of genes.
Suggest they're in order, yeah.
I like that better. Okay. So a chromosome is a linear sequence of genes. A gene is a linear sequence of nucleotide bases, right? Linear sequence of nucleotide bases. So now, what about a mutation? Let's do a mutation. A mutation is a change in some nucleotide bases, you know, in a gene, some change of one or more nucleotide bases, a change in one or more nucleotide bases in a gene that, now I'm making my own little definition here. You read it in a book or a textbook or a dictionary, it might say it a tiny bit differently, but you'll see why I'm saying it the way I'm saying it in a second. Change in one or more nucleotide bases that might affect the amino acid that's coded for it. Amino acid or acids. So I'll make a singular or plural depending on how many bases are changed. So it's a change in one or more nucleotide bases in a gene that might affect the amino acid it's coded for. Might. That's kind of a funny word. Why might?
Because it might not.
Does change. Well, here's the thing. Let's just say it's what they call a point mutation. So let's use this word. Point mutation. I'll put it in blue again. So a point mutation is one nucleotide base change. I'll say a change in one nucleotide base. Now, how many nucleotide bases will make up a gene? Depends on how big the protein is going to be. It could be thousands of nucleotide bases. We're only talking about one change. Is that going to change what amino acid is being coded for? It depends on what the change is. I mean, you know, the nucleotide bases. Now look at this chart. Let's look at this chart a second. The nucleotide bases, you know, the three of them, and they're read three at a time, right? And that's called, what's the term for three?
Codon.
Codon. Good. They're read three at a time. But look at this. Look at what if you had the codon in this one particular mutation. Let's say the original one was CUU. We'll say CUU. It codes for isoleucine, right? CUU. Look at CUU. Isoleucine. What if that U got changed to a C? It'd be CUC. It'd still be isoleucine. What if it got changed to an A? CUA. It'd still be isoleucine. So if it's the third base, in some cases it may not change it. In other cases it will change it. If it's the first or second base, it's more likely to change it. But it's it's not. In other words, it's not an absolute. So the mutation could be neutral, meaning it doesn't even change the amino acid. I said it changed the amino acid, so that was a little bit not stated correctly. I said it might affect. That was okay saying it that way because it may not. So we'll say a mutation. So a mutation might be neutral. Because even though there's a change in nucleotide base, it might still code for the same amino acid, especially if it's the third base of a codon. In a lot of those cases, it won't change the amino acid. The mutation might be neutral. No change in the amino acid codon for. Okay. So it's no big deal. It changed codon slightly, but didn't change amino acid. Something I didn't get into, but of all these amino acids, some of them are acidic, some are basic, some are hydrophilic. Some now I say these amino acids, but I'm really talking about the side chain. Some are hydrophilic, some are hydrophobic. I mean, there's all kinds of possibilities. And it could change the amino acid, but it might be a very similar amino acid, so it may not affect the protein. So it could be neutral. It could be neutral. It could be only slight change. One. The mutation might be. I'll say 1 neutral. I'll say 2 only a slight change. And I'll say similar amino acid, not the same amino acid, but similar. I'm going to put AA for amino acid. Could I just do that to shorten?
Yes.
I have to write amino acid every time. A similar amino acid coded for, or could code for a totally different amino acid, had totally no relationship. So it could be major change. Different amino acid. Different amino acid with different properties. And then, for example, could be, I say, hydrophobic side chain versus hydrophilic or something like that. Now I'm going to give you an example. Can you read this? No, you can read this. So I said it could be a major change, meaning it's coding for a different amino acid, and that amino acid has very different properties from the original. Question? I thought that.
I did.
Okay.
Yeah, I was just. Those are like the 3 types of mutations.
I mean, I'm just saying that mutations, in terms. What I'm trying to get across here is that mutations are always bad.
Mm-hmm.
Well, and actually, well, okay. I don't want you to think just because it's a mutation is bad. Sometimes it's totally neutral, it doesn't make any difference at all. Sometimes it's totally bad. Sometimes it might even be cause an advantage. See, okay? So let me give you an example of one little point mutation that turns out to be bad. Yeah, let me explain.
Amino acids change like that can cause like physical differences.
Okay, well, so let me give you an example. This is a good example of a major change, just from one point mutation. And it has to do with hemoglobin. Hemoglobin carries our oxygen in our red blood cells.
Like sickle cell or
Sickle cell is what we're going to talk about. Thalassemia. These are two.
But doesn't that have to do with the different
Well, let me tell
blood
but
typing?
no, not blood typing. This is strictly hemoglobin.
Yeah, but it also only affects a certain racial.
No.
Or it's more common.
It's more common. These are hemoglobin changes, and they turn both thalassemia. The word thalassa means sea in Greek, and it's because it's common in the Mediterranean, mainly around the southern Italian and the Greek communities. And sickle cell anemia is more common in the West Congo region of Africa. Okay, now these are, each one is a point mutation. They're slightly different in two different anemias.
But
So
wasn't
point
the
mutation. Well, let me tell you. I'll answer your question. I'm pretty sure. Wait. One point mutation which changed one amino acid, and it turns out for sickle cell anemia, out of 100 and some odd, no, I don't know how many amino acids in the thing, it's the 6th amino acid. One little change in amino acid, but that can cause a difference in the way the molecule forms a glob, you know, the way it folds. It changes the way it folds in low oxygen tension. Okay. Now, that can be a problem. If you have 2 genes for sickle cell anemia, you won't survive. Okay. But if you have 1 gene and you're a carrier, you can have problems. And they call it sickle cell crises. And in low oxygen tension, these hemoglobin molecules, they don't form the normal shape. They'll change their shape. It'll have an effect on the cell. The cell becomes sickle cell, you know, shaped like a sickle, you know, kind of like this instead of nice and flexible. Gets caught in things like your capillaries, and it can cause lack of blood to various things. They call them sickle cell crises. They can be deadly. And you're going, why would there be a gene that has the potential of being deadly that's being perpetuated in the, you know, in the gene population, gene pool as we call it? But the 2 areas, and the thalassemia works in a similar kind of way. But the 2 areas where these anemias occur are where there was a lot of malaria. And if you are a carrier, not if you have 2 bad genes, if you have 2 sickle cell genes, you won't survive. But if you have 1 sickle cell gene, 1 normal gene, you are actually protected against malaria. You have a better chance of surviving malaria. And by the way, malaria still kills like between 10 and 100 million people a year in the world. I mean, up to 100 million people a year are still dying of malaria. I mean, this is serious. You might have read a year or so ago
in Panama.
Microsoft, Bill Gates, his foundation is trying to put a lot of money into malaria research because they're hoping to try to fight this. But anyway, you can see that if you're in an area where you have a reasonably high chance of acquiring malaria and dying, and yet if you suddenly carry a gene that in normal circumstances might not be good, but in these circumstances where you're around malaria, it gives you protection and you can survive, you can see how that gene would survive in the environment. Or the gene pool, I should say.
It's beneficial.
Again, it's not, you know, in general, in normal situation, you say that's not a good gene. But in the situation where you're in the presence of malaria, it would be advantageous having 1 copy, not 2 copies. And it's 1 point mutation. That's all it is. 1 little mutation in 1 nucleotide, which changes 1 amino acid in that hemoglobin. And yet it can have a profound change in, you know, in this case, survival if you're around malaria. So it's kind of a wild thing. Yeah.
I was just curious, what about CJD? How is that a mutation?
Which one? The
Creutzfeldt?
Creutzfeldt-Jakob? All right. I don't want to get into all this, but Creutzfeldt-Jakob, or Jakob disease, is a weird one, which they now think is caused by a prion. Now, prions are little strange little characters. The guy who discovered prions, and his name is Stan Prusner, and I met him. I wouldn't say I know him, but I met him. Hugh Gigo, good researcher, and he did get the Nobel Prize. He's at UC Med Center. I think he's still there. There's still some questions about prions, and I still have some questions about prions. He's probably absolutely right with what he says, but I myself still have some questions about how prions work. And so they think that's what causes, you know, they now think that's what the cause of Creutzfeldt-Jakob disease.
Well, they're saying that it can be genetic.
Yeah, maybe. I mean, that's what they used to think. Now they're not so sure because of these prions. So I still have some questions about prions, but he did get the Nobel Prize. He's done some good research. I knew the gal who worked in his lab, and that's how I, met him. And that, he's probably absolutely right. But I myself still have some doubts about it. So other than that, I can't say. I don't.
Skepticism is good.
But anyway, back to this. So here's the thing. In the case of that little point mutation, for normal hemoglobin to become sickle cell hemoglobin, it caused a profound difference. In this case, ordinarily bad, although it might be advantageous, if you were around malaria. But a mutation could actually have a good effect. It may be a major change, number 3, but it may actually be advantageous. It may actually give you survival. Well, I get that gap I gave you, survival ability. But it could be some other kind of a difference, and it could give you some survival advantage too. So mutation isn't always bad. But here's the thing to think about. Since organisms on Earth are pretty much fit for their environment, because of all these thousands and millions of years of evolution, most changes will be for the negative,
Yeah.
Only an occasional change will be positive, will be useful. So you can figure that in general, if there's a mutation occurring, you can figure it's a good chance it could be a bad thing. If it's neutral, it doesn't matter. It doesn't make any difference one way or the other. If it's the second kind where it's a slight change but it doesn't change the actual changes amino acid, but it's a very similar amino acid, probably no big deal. But if it's a major change, probably it's not for the better. But still, mutations do provide more variability in the environment, in the gene pool. And the more variability in general, the better. Because, let me give you an example of what I'm saying here. Cheetahs, cheetahs, they're very fast runners, right? There is not a single wild cheetah in the world. All cheetahs are in some kind of control. They're either in some kind of a zoo situation, there's some kind of a big park situation, whatever, but there are no free-ranging cheetahs. And the other thing about the cheetahs, that's not the big problem, but the big problem, except for what I'm getting to now, is that the gene pool is very limited in the cheetah population. It's not a very big gene pool, meaning there's not a lot of variability. If there's not a lot of variability, if something came along, like some disease, some virus that was particularly virulent, deadly, it could wipe out almost the entire population of cheetahs. Whereas if you have a much more variability in a population and something comes along, there's better chances there are going to be a few individuals who can survive it.
Mm-hmm.
And so that's why they always worry. They always worry about a population if the gene pool doesn't have much variability. So in general, we think of variability being good because it provides more possibilities of
dealing with stuff,
withstanding whatever the environmental insults might be. Another thing, geneticists and others worry about what we eat. We eat what? We eat soybeans, we eat wheat, we eat corn, and everything. But most of those are what they call monocultures. They're all pretty much same species. And if some blight comes along, some fungus or something that destroys these things, it can destroy this huge crop of, say, wheat or whatever, and it's a major food thing we eat, and it'd be gone. Or a huge percentage of it would be wiped out. So in general, biologists say we want variability in the population. That's a good thing. So anyway, so mutations do provide variability. So therefore, they are providing, for evolutionary purposes, they're providing this variability, helping to allow, this survival of, what we used to call the survival of the fittest. It's a little more complicated than that. But anyway, all right. Does that make sense what I'm saying? So mutations, again, don't think of it as absolutely negative. But more often than not, it probably is. It's not an absolute. All right, let's talk about cell cycle and all that. See, a few more terms that we're going to get to. Actually, this lecture on mitosis is supposed to go today and Wednesday, so I have plenty of time, but we need to be really careful when we're talking mitosis to get the terminology right and you're going to see there's a lot of words, and we're going to use them and use them and use them in certain specific ways, and it's kind of important. So let me write down some of these terms. Let's see. Did I ever give you the word genome? Let me just give you that word. I've used the word, so let me give you the word genome. genome just refers to all the genes in a population. That's the gene pool. The genome is all the genes in an individual.
Being species
But
specific,
if we
you
say
mean?
gene pool, yeah, usually. I'll say in species. Yeah. And if I say gene pool, that's all the genes. See, this is okay. There's a lot of little subtleties here that aren't that critical, but if I talk about the human genome, that's all the genes in the human. But keep in mind that whereas we probably share 95% of our
genomes, yeah.
we have changes in at least 5%. We have variability. You know, you'll have one copy, I'll have one copy, you'll have one copy, and that's why we can't just do simple organ transplants. You can't just transplant a heart or a kidney or something simply to one person or another because we've got some differences and the body's going to reject it. And only if you happen to have an identical twin and you needed a heart transplant and they wanted to give up their heart to you, which somehow doesn't seem likely, otherwise, the heart's going to be, you're going to reject it, and so then you have to take all kinds of drugs to try to inhibit your rejection process, to inhibit your immune response. So when we talk about the gene pool, that's all the genes, So this is all the genes, in all the individuals. In a species, say. So there's a little bit of overlap in what I just said, because we'll talk about the human genome, and I said they sequence the human genome. Whose DNA did they use? I don't know actually, but they only used one person or maybe several persons, And if you sequence your genome and my genome, as I said, we'd have some differences. But if we took all the differences from all of us, as I said, 95% or so of the genes will share. Which of all the differences, that would be the gene pool. And so when I used that word a few minutes ago when I was talking about sickle cell anemia, why would that sickle cell gene or the thalassemia gene remain in the gene pool if it's a bad gene? Well, it has, as I said, some ability to give you protection in certain environments. Anyway, okay. So these terms are not quite interchangeable, but they're pretty close. Some other terms, let's see, that we haven't talked about. I don't know. Yeah, that's enough. Okay, let's talk about terms that are going to get us to understand mitosis. First of all, what the heck is mitosis? Let's talk about mitosis. Just a word, just what we're talking about. And when I talk about this, I want you to think about biology, and for the last over 2,000 years, biology has been mostly observational, almost totally observational. And I say over 2,000 years because one of the best biologists of all time was Aristotle. He was a brilliant observer and a brilliant. He was a philosopher, as you know, and all that. But he was a good scientist. I mean, he made errors, obviously.
Is he the one that came up with the
a heavy object and a light object? The heavy object would fall faster, and people believed that for years because he said it. It turns out they don't. They fall at the same speed. At least in a vacuum, they do. But he also made some brilliant observations in biology. For example, he talked about a blind porpoise. People thought he was mistaken. It wasn't until about 100 years ago or something they discovered the porpoise he was talking about. It lives in, or dolphin, it lives in India. And he had described it. And his description was exactly correct, They just didn't see it for a long time. So he was really a very good biologist. But anyway, the point is that, and I've said this before to you, biology has been observational. Because you see with the naked eye. Once they invented the microscope, they could see little tiny things, right? Things you can't see with the naked eye. So that opened up a whole new world. So that was in the 1600s when they invented the microscope, but they really perfected it over the next couple of hundred years. As I mentioned to you, by mid-1800s, the microscopes they made then were as good as the microscopes we make now. We have better lighting conditions now.
Even
We also have
electrons?
different kinds of microscopes they didn't have then electron microscopes and scanning electron microscopes.
Yeah.
And, blah blah blah. There's all these other ultraviolet microscopes and you name it. We have all kinds of different sorts of microscopes. But the regular light microscopes that we're using in the labs over here, the labs, they've had as good scopes as we have for the last over 100 years, 150 years. But biology was observational. It's only really been since the 1950s that it's been mainly, maybe 50% or more experimental. So biology really joined the hard sciences, physics and chemistry, and became more experimental since the 1950s because they've got such good techniques. But anyway, back to the observations. So people were looking at cells under the microscope. The cell theory, came about in the mid-1800s, 1860s or so. The cell theory came about. So since then, people have been looking at cells and trying to make sense out of them. And you look at a cell. Now you're looking at dead cells most of the time, not always, but most of the time you're looking at dead cells because you're taking something and you're putting stains on it, looking at it under the microscope. You look at a cell and not much is happening. And then suddenly you start looking at certain cells and you say, oh, hey, something's happening in the nucleus. Well, we call it mitosis. But they get, oh, yes, God, we're getting something happening. We call it mitosis. So they made a big deal about it. And so they talk about the cell cycle. And they say, okay, there's interphase. And then there's mitosis. And if you take a mammalian cell, just say a typical mammalian cell, on the average, there's no magic here, but the interphase lasts about 23 hours, and mitosis is about an hour, about 1 hour, and so maybe half an hour, but let's say an hour. And so they said, okay, there's interphase followed by mitosis. And now, mitosis really isn't cell division; it's nuclear division, and I'm going to make a point about that. But we will, in a sloppy way, just use the word mitosis to mean cell division. But usually, almost always, but not always, nuclear division is followed by cell division. But anyway, here's the point. So they said, okay, the cell's not doing much for 23 hours, and so oh God, we've got something happening. This is great. Well, of course, the cell's doing something in interphase. That's when it's doing all its normal cellular activities. It's just that you couldn't see it when you're looking at the microscope, Once they started doing experiments, of course, and they know all kinds of things that is happening in mitosis. But until they did these experiments, they said, not much is happening, which is kind of ridiculous because the cells aren't just sitting there. I mean, as I said to you before, if anything happens, your body is happening because some cell's doing some work. And how can cells do work? What are the worker molecules? Oh yeah, I just want to make sure you remember that. Okay, proteins, of course. Yeah, but anyway, okay. So let's just actually talk about what these words mean. So mitosis, what mitosis means, and then I'll give you more detail on the cell cycle. Mitosis means nuclear division. And this is almost always, or not always, but almost always, I'll say usually, followed by cell division, which, and the proper term for cell division is cytokinesis.
Nuclear division is followed by.
I'm sorry, say it one more time.
Nuclear division is followed by cell division.
Yeah, nuclear division, which is usually followed by cell division. So, I just put up, what I put up there, interphase and mitosis. That was sort of what you'd say in the old days. We still use the word interphase, but in the old days they said, oh, interphase is the time in between mitoses. Okay, well, interphase is when most everything's happening. But if the cell's going to divide, and we're talking eukaryotic cells, it's got to go through this mitosis, which is nuclear division. So what are we talking about here? What are we talking about? So let's actually look at the cell cycle in more detail. So the cell cycle, now they call it G1 phase, S phase, and the G2 phase. And then we'd follow this by mitosis and then cytokinesis. And I'm going to say old idea up here, where I just had interphase and mitosis, and we'll say this is more the new idea, where we have, interphase, which would be these three phases here. This would be interphase, G1, S, G2. We say, hey, something's happening there. First of all, G1, G stands for gap. And they said, hey, this is just the space between what's happening here versus what's happening here. In the case of G1, it would be from cell division until synthesis, which is what S stands for. And then the time between synthesis and mitosis was gap 2. It's kind of weird. But so G1 stands for gap. I say G equals gap. And S equals synthesis. And then of course G2 gap again. But of course normal activities are happening. Cells are doing work. There's all kinds of chemical reactions going on at any one moment. So what's happening during G1 phase? Normal cellular activities. Whatever they might be, depending on the cell. It's the same thing for G2, normal cellular activity. But see, they used to think of the cell was just sitting there doing nothing until it divided. Well, that's kind of ridiculous. And if in cells that stop dividing, like your muscle cells are never dividing, ever again. They say those are in a state of G0. And the old term for it was a resting phase. They're not resting, they're just not going to divide. They're still doing their normal cellular activities, meaning they're busy. If they're a muscle cell, they're contracting when you tell them to contract. So you have to be careful of things like using the word resting phase. It's not resting, it's working. At any rate, synthesis. This is what we're interested in for us today. Synthesis. This S phase, that's when all the chromosomes are duplicated or replicated. You can say duplicated, but as I told you, replicated is usually the word we use.
Could be done more than once.
Now, that's not only the chromosomes that are replicating. It's not only making synthesis, isn't only a new DNA, but that is what we're talking about here. But also all kinds of proteins are being synthesized and replicated. The spindle apparatus, the spindle, it just went out of my mind. It'll come back. Those little things are replicating. So all kinds of things are replicating. But what I'm interested in for us is the chromosomes being replicated. But there's also synthesis of all kinds of other things, proteins and everything else. So if a cell is going to divide, and some of our cells, like our skin cells, are constantly growing, dividing, this constant thing. They're constantly going from G1 to S to G2 to mitosis to cytokinesis, back to G1, just constantly happening. On the average for a mammalian cell, it's about a 24-hour thing. There's nothing magic. It's not every day that they happen. It may be more rapidly or less rapidly. Some cells, like your liver cells, they're not going through growth and division. They're just sitting there doing their job. They're very good at doing their job. They're very busy doing their job. If you damage those cells, then that'll stimulate the neighbor cells to start growing and dividing. So they'll suddenly come out of their quote resting stage, which is not resting, but that's what they call it, and start going. And they'll sometimes what they'll do, they'll say that if a cell like the liver is not going to divide, they'll say, okay, from G1 you could go into G0, quote resting stage. which the cell isn't divided. So in general, your liver, your liver cells are in what we call G0 because they're not dividing. But if you damage the liver, then the nearby cells will be stimulated to grow and divide and fill in the gap where you lost the cells. So these guys would could come back to S phase. The liver cells can do that. But if you take something like your muscle cells or your nerve cells, your neurons, they can't do that. They're in G0. They're still doing their work. They're working hard, but if you damage them, they'll die. They won't be able to, divide. So you lose muscle cells, you don't have as many muscle cells. You lose nerve cells, you don't have as many nerve cells. Now they say when you get to be older that you're losing X number of nerve cells every day. Maybe.
Yeah.
Probably true. No, most of the things that they say, by the way, these things are myths. I'm not saying I haven't lost nerve cells. I probably have, but I don't think I've lost nearly as many as
Neurons
they used to
regenerate,
say, because when they did their experiments on people, not experiments, sorry, you're not supposed to experiment on people. When they did their studies on people, they would study dead people, cadavers. But if they were talking about young people versus old people, usually a young person dies because of some problem, like they may have had some disease or they may have, overdosed or something like that. And they may have damaged things like nerve cells, And so you look at that, and then you look at an old person who died of old age, Anyway, they made a lot of predictions that weren't necessarily accurate because they weren't really comparing the same kind of a population. So when you read about, you lose 35,000 neurons every day, I don't think that's true, and I don't even know what numbers they give.
So it's kind of like the standing on the flat Earth before
Yeah,
the reason Heisenberg is wrong.
Right. The reason he's wrong
is because he's the
uncertainty.
He's uncertainty principle. He wasn't certain about whether or not we'd ever be able to measure electrons. And now I found out. I thought of it as soon as I heard Heisenberg principle, and I'm like, I got this guy beat. I'm like, why didn't Einstein think of it? And then I found out, somebody else just thought of it 5 years ago. So yeah, still 100 years or not 100, but 50 years to figure out he's wrong. I'm like,
yeah. So don't take a lot. Some of these things you'll hear myths that they're exaggerations. Don't take them literally. But I'm getting off the topic. I want to get back here. But go on.
On the Heisenberg principle. Oh, doesn't it state that you can't predict
where
an electron
he says you can't. He says if
There's
you're
no
trying to find out where an electron is,
because you affect it when you measure it, but we don't do that
can't.
now.
See, they want to know its location and its momentum, and you can't. If you find his location, you affect his momentum, and vice versa. So you can't, because your way of measuring it is going to affect what it's doing.
But
anyway, we don't want to get into that. That's not us. That's physics. Yeah.
Even though no new nerve cells are made,
Right.
Aren't neurons built like throughout your life?
Okay, now, so I just said that neurons and muscle cells can't ever divide, and that's not absolute because certain ones can, but other ones they can't, and they'd like to learn how to get the ones that can't to divide. If they can, they can do some repair of people with various problems like Alzheimer's and various other things that can happen. But there are certain ones that do and can grow and divide, like the ones for your sense of smell. But most neurons, they can't. At least we can't. We don't know how to get them to, and they don't do it normally. But anyway, so the point is that this whole thing that you'll read about, this resting state or G0, what that says is the cell's not going into division. He's staying in what we would call G1, except that they just call it G0. Some cells, if stimulated properly, can go through the division, but other cells, like the muscle cells and stuff, can't. But that's not important stuff right here, so I'm going to take this off. Good. So now we're back here. Now, here's the deal. First of all, when DNA is replicated, it's very accurate. And I mentioned this to you last time that they use terms like, if you are proofreading on a paper to see if you made any spelling errors, it's the same kind of thing. They talk about proofreading when DNA is being synthesized. And they talk about the proofreading enzyme and copy errors. Now they'll remove copy errors and all that. So DNA replication is incredibly accurate. Occasional mistakes, but they're rare, very rare. Like they figured like there's maybe one mistake out of every 3 million bases that is put in. 3 million nucleotide bases, God, that's a million codons. I don't know, that's many genes worth. So that's pretty accurate copying. But let's talk a little bit about. We're not going to talk about the actual copying, but just what happens. So because this is what we're interested in right here. Is when the chromosomes are replicated. Now we said we, meaning I was saying, you think humans, but eukaryotic cells, we have pairs of linear chromosomes, right? Okay. So when the chromosomes are replicated, we'll do this. This is an S phase, right? So when the chromosomes are replicated, now here we're going to get into some terms, and you've got to be real careful on the terms. Once you get the terms straight, then it'll make understanding the words that I'm using, or the books are using, you make that understanding very easy. But until you get the terms right, you're wait, what are you talking about? Chromatids, chromosomes? We'll get that. When the chromosomes are replicated, then there are 2 copies. There are 2. Let me do this. Let's talk about each. When each chromosome is replicated, let's do it singular, is replicated, what do you have? You have then 2 copies that are actually identical, right? They're 2 identical copies. They're held together. And as long as they're held together, they're going to be called, this is the term they use, sister chromatids. Okay. So when each chromosome is replicated, there are 2 identical copies. Of the chromosome. And they are held together. They're joined. And as long as they're joined, we don't call them 2 separate chromosomes. They give a different term. They say that's 1 chromosome made of 2 identical sister chromatids. And they're 2 identical copies, and they are joined. And when as long as they're joined, these 2 identical copies are called identical sister chromatids. So as long as they are joined, these 2 copies, these 2 identical copies, are called identical sister chromatids. By the way, it has nothing to do with sisters or brothers or anything. This is a term. It's just again a term. So we have a parent cell, and then when that parent cell is going to divide into 2 separate cells, those are going to be called daughter cells. It's just a term they give. It has nothing to do with sons or daughters or anything else. It's just words that they use. All right. So when each chromosome is replicated, you have these 2 identical copies, and they're joined together. And they're called identical sister chromatids. All right, so you've got to be real careful on this. When they separate during mitosis, when they actually are separated, then we'll call them chromosomes again. But as long as they're joined, they're going to be called sister chromatids. All right, you see that? That's an important concept. And you're going, why are they making a big deal of it? It has to do with the number of chromosomes per cell. They can say, hey, we always have the same number of chromosomes in a given cell. And it's terminology. That's really the thing it is. All right, now another thing we need to worry about, we're talking about eukaryotic cells, right? And what do we say about eukaryotic cells? We have pairs
23.3.
of linear chromosomes. Well, humans have 23 pairs, right? Pairs of linear chromosomes. So let's write this up. So eukaryotic cells, we had it up earlier, but I'm going to do it again. Eukaryotic cells have pairs of linear chromosomes. So, for example, humans have 46 chromosomes for 23 pairs. And, you know, they're given numbers. Well, the sex chromosomes are called X and Y. We'll get to those later. But all the others are called chromosome number 1, chromosome number 2, chromosome number 3, all the way up to chromosome number 1 and 2. But we have 2 of each. We have 2 number 1s, 2 number 2s, 2 number 3s, etc., etc., etc. Okay? So we'll say 2 number 1 chromosome. 2 number 2 chromosome, 2 number 3 chromosome, etc. Okay? Now I'm building up to another term that's important. And of these 2, you know, these pairs, 1 member of each pair came from your mom, 1 member of each pair came from your dad. So if we're talking about 2 number 1s, 1 number 1 from mom, 1 number 1 from dad. You know, and then we say, oh, here, 1 number 2 from mom, 1 number 2 from dad, etc. Right? Just again, I'm belaboring this on purpose to make it sink in. So, new term, it's an important term, and you'll need it for this lecture and 2 more lectures, is the 2 number 1 chromosomes are said to be homologous chromosomes. The 2 number 2s are said to be homologous. I'm going to write it down. The 2 number 3s are said to be homologous. So, so I'm going to write this. So the 2 number 1 chromosomes, I guess I can do this, would be homologous. As a homologous pair. Chromosomes. These would be a homologous pair of chromosomes. These would be a homologous pair of chromosomes, etc. So what's the deal? What are we talking about? The 2 number 1s. You get a number 1 from your mom and number 1 from your dad. What's a chromosome? It's this linear sequence of genes, right? The linear sequence of genes. And the genes the genes on your mom's number 1, the genes on your dad's number 1, they're almost the same. I mean, you know, as I said, maybe 95% of them are identical. The other 5%, there are a few changes here and there. But that's the variation. But they're the same kind of gene. They're coding for the same kind of protein. The protein may be slightly different. You might have brown eyes versus blue eyes versus green eyes, whatever. You might have brown hair versus whatever. You know, you see what I'm saying? You might have slight differences in what the actual outcome of the gene is. But, you know, you'll have a gene for your eye color, say. Probably more than one gene. I mean, you have pairs of genes, but I mean, you probably have more than one pair for eye color. But let's just make it simple and say you have one pair. You got one of those genes from your mom, one from your dad. The way it comes out is whatever comes out. And the 2 genes you got from your mom and dad may be slightly different. One may be blue eyes, one may be brown. But they're still coding for eye color. You know, you see what I'm saying? Does that make sense? So the number 1 you got from your mom, the number 1 you got from your dad, they're said to be a homologous pair because they're coding for the same kinds of proteins. The number 2 you got from your mom, the number 2 you got from your dad, homologous chromosomes. Okay?
Do you have any idea why eyes would change color? Like, because when I was born, I had blue eyes, but now I have green eyes. When I was 3, they changed.
Well, you know, they usually change when you're born to when you're like several years old, but usually they'll change within the first couple of years. But this one pediatrician that my kids had said that he had a girlfriend whose eyes changed color in college. It was that late. Why? I don't know exactly what's happening. Something about the expression of the genes,
That's
but I don't know why exactly that happens. And so, yeah, I mean, it's getting into regulation of that expression, which I can't.
would it be like a time delay in your genetics or something like?
Something,
I mean,
yeah,
because
yeah,
I had red
something.
hair when I was a baby. By the time I was 8, it was brown. But
Yeah, and again, that's common where you're expressing more of the color as you get older for reasons that until you lose it, until you lose the color. Yeah.
Can you define homologous?
Yeah, well, so homologous, in other words, when let's say the number one you got from your mom,
same information.
when it replicated, they're actually identical. So you have 2 identical sister chromatids. But the number 1 chromosome you got from your mom, the number 1 you got from your dad, they're coding for the same kinds of proteins. There might be slight variations. And that's why I'm saying maybe blue eyes versus brown eyes as far as whatever the enzymes are involved. So they're said to be homologous because they're coding for the same proteins. They're not identical because no 2 people have identical chromosomes except identical twins. Does that make sense?
Yeah.
So homologous just means that they're coding for the same proteins. It's an important word. It's going to be especially important when we get to meiosis, but it's especially the kind of cell division that is used to make, or nuclear division, cell division, used to make eggs and sperm. This is just normal nuclear division followed by cell division for cells that are just growing, dividing all over the place. Okay, so far.
Mm-hmm.
So the 2 important words here are these things that we call identical sister chromatids. And as long as those 2 identical chromosomes are held together, or identical, we'll say chromosomes, we'll call them sister chromatids. And then each pair that you got from, the pair, 2 number 1, 2 number 2, each one of those pairs is called homologous chromosomes. Okay, now we have enough time to give you the introduction, and then I'll go through the details on Wednesday. But you're going to do this in lab also. We can talk about this in lab. So it's going to help. I think it'll sink in a lot by doing it both in lecture and lab. So let me tell you something about, again, mitosis. Let's talk about what is the idea of mitosis. What happens in mitosis? I mean, what happens after mitosis? I'm just erasing it, but what is it?
Cytokinesis.
Cytokinesis, which is
action in cell
division. So what we're talking about is the process in which cells are going to divide.
So that means the nucleus divides and then the cell divides?
Exactly.
Okay, that makes sense.
And usually right afterwards. There are times when it doesn't happen. You get mitosis, and later it's got cell division, a whole bunch of cell division, but that's usually an embryonic development or something.
Oh, okay. No, I get it.
Normal circumstances.
When we're not embryos, it's not a big deal.
So, anyway, but here's the deal. When a cell's going to divide, you have some parent cell, and what we would, quote, want is that when that parent cell divides, you get 2 identical daughter cells. By identical, we mean they have the same genetic information. Identical genetic information. So we'll say when a cell divides, this would be the parent cell. In fact, I use the word parent cell. You don't have to use the word parent cell to say a cell, but technically it's a parent cell. So when a parent cell divides, and this is happening when you're a little kid. Just the word parent doesn't have anything to do with adult. When a parent cell divides, I'm going to say we want, and I'm going to put it like this, because obviously there's no desire going on here. This is what happens. We want 2 identical daughter cells. And identical in this case meaning with respect to the genetic information, the DNA. So, identical with respect to the chromosomal material, or the chromosomes. I'll say chromosomal information.
This kind of when the cells divide from a parent cell and the daughter cells are smaller and then they grow into the size of a normal cell. Is that what's happening?
Exactly.
Okay.
And there seems to be some, for most cells, there seems to be some kind of maximum size. You get to be too big, then the cell needs to divide. And the reason, if you think about it, and this is not something you need to worry about for the class, but as the cell's getting larger, you have to get more membrane material, right? That's fine. But what's that membrane? Remember, I made a big deal about how important that membrane is because this what allows things in and out.
With the phospholipid bilayer?
But if we're talking, but that's what lets the phospholipids and proteins are what let things in and out, right?
Mm-hmm.
But as the cell the cell grows, and we're talking the surface area of the cell membrane. Surface area, that's square, right? Surface area of a sphere. That's a squared function, whereas inside is the volume. The volume is a cubic function. So in other words, the surface area increases by the square of the radius, and the volume increases by the cube of the radius. So the volume increases a lot faster.
Mm-hmm.
But why is that a big deal? Why am I, the surface area is where you get things in and out, like nutrients coming in and getting rid of waste products. But your metabolism depends upon your volume. So if your volume is increasing real fast, because you've got to be real large, the cell's got to be real large, so you've got a lot of volume, so a lot of metabolism going on, you may not be able to get nutrients in and waste products out fast enough if the cell's too big. So for practical purposes, you can think of a cell when it gets too big, it needs to divide. If you want to put it that way.
I get it.
That's really kind of
that makes sense.
Anyway, there was a movie called The Blob, I think it was, or something like that.
Oh yeah.
And I think there was some cell that just was eating everything inside and getting bigger and bigger, and I think it eventually ended up engulfing New York City.
Yeah.
It seemed like a
it was an awesome movie.
Anyway, I'm kidding. Okay. So anyway, so in general, parents, when cells are growing, they'll grow and divide, they'll grow and divide. But when you divide, you want these two identical daughter cells with respect to the chromosomal information. So mitosis, what mitosis does is ensures that the replicated chromosomes are equally distributed, so that each, those sister chromatids, one sister chromatid of the number one will go to one new daughter cell and one will go to the other daughter cell, one of the number two. So ensures that the replicated chromosomes meaning the identical sister chromatids, identical sister chromatids will be separated and one of the chromatids goes to one cell, the other goes to the other cell of the new cells. And one sister chromatid, so now we'll call it a chromosome, one sister chromatid will go to one daughter cell, and the other to the other one. In other words, this is an elaborate process that makes sure that each daughter cell gets a complete set of the chromosomes that the original parent cell had. They'll end up with two number ones, two number twos, two number threes. But, one of the number ones came from the dad, one of the number ones came from mom. But it has a complete set. So it's an elaborate process. Now, since we can't really start, let me just give you the terms for it. just so you'll have it in your mind, and then you're going to do your lab. If you do your lab, some of you half of you do it tomorrow. The other half you'll see me on Wednesday, and then you'll do it Thursday. But the mitosis has it's four steps, and again these are things that could be observed, or phases we should say, 4 phases or phases. But these are things that can be observed in the microscope. Keep in mind that this is a continuum. It starts and just keeps on going to the end, But we're looking at it in a microscope where it's not happening at the moment it's been stopped, and you're looking at it. So we've, over these decades, people who observed this, came up with these 4 stages. I said phases, stages, oh, whatever. Stages, phases, stages. Oh, call it whatever you want here. I don't care. Okay, so it's prophase. So you had the phase in here. Metaphase, anaphase, and telophase. Now, the way your lab manual tells you to remember to
Pros first,
PMAT.
not as big.
PMAT. Oops, sorry. If you just learn PMAT, then it's prophase, metaphase, anaphase, telophase. Whatever you want to do to remember it. As I told you, I just memorized it when I was, originally way back in 100 years ago. But PMAT is good if that helps you remember whatever. On Wednesday, we'll go through the various phases, and talk about what's happening in each one. In the lab, you're going to look at the root tip of an onion, and the root tip is growing rapidly. They made these little thin slices. You can look at that, and you can actually identify the stages. And if you're going to have it tonight or tomorrow, rather, you can look at it in the microscope. They'll go over to class and you'll see these things. There's whitefish, blastula, which is just not too long after it's been fertilized, and again growing, dividing. So you'll see a whole bunch of mitoses occurring. And the word they use for that is mitotic figures. It's a funny little term, but that's the word they use. You're looking at mitotic figures, and when you're looking at a mitotic figure, it can be any one of these stages. And when you think about that, a typical cell, mammalian cell, now I don't know what it is for the plant, but the typical mammalian cell spends like 23 hours in interphase and 1 hour in mitosis. So when you're looking at these cells, the majority of them are in interphase, but a few of them are going through mitosis, and those are you'll see the so-called mitotic figures. Which phase will they be in? Just chance, whatever they happen to be when they made the slide. But if you look around, you'll actually see them. So we'll talk about each phase and what's happening on Wednesday, and you look at them in the microscopes in the lab. Okay?
All right.
Make sense?
Cool.
All right. See you guys Wednesday.
All right.